Photograph a historic site, help Wikipedia, and win a prize. Participate in the world's largest photography competition this month. Learn more. Cytoplasm. 86 languages. Cell biology. Animal cell diagram. Components of a typical animal cell. 1 nucleolus. 2 nucleus. 3 ribosome, dots as part of 5. 4 vesicle. 5 rough endoplasmic reticulum. 6 Golgi apparatus, or Golgi body. 7 cytoskeleton. 8 smooth endoplasmic reticulum. 9 mitochondrion. 10 vacuole. 11 cytosol, fluid that contains organelles, with which comprises cytoplasm. 12 lysosome. 13 centrosome. 14 cell membrane. In cell biology, the cytoplasm describes all material within a eukaryotic cell, enclosed by the cell membrane, except for the cell nucleus. The material inside the nucleus and contained within the nuclear membrane is termed the nucleoplasm. The main components of the cytoplasm are the cytosol, a gel-like substance, the organelles, the cell's internal structures, and various cytoplasmic inclusions. The cytoplasm is about 80% water and is usually colorless. 1. The submicroscopic ground cell substance, or cytoplasmic matrix, that remains after the exclusion of the cell organelles and particles is ground plasm. It is the hyaloplasm of light microscopy, a highly complex polyphasic system in which all resolvable cytoplasmic elements are suspended, including the larger organelles such as the ribosomes, mitochondria, plant plastids, lipid droplets, and vacuoles. Many cellular activities take place within the cytoplasm, such as many metabolic pathways, including glycolysis, photosynthesis, and processes such as cell division. The concentrated inner area is called the endoplasm and the outer layer is called the cell cortex, or ectoplasm. Movement of calcium ions in and out of the cytoplasm is a signaling activity for metabolic processes. 2. In plants, movement of the cytoplasm around vacuoles is known as cytoplasmic streaming. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. History, edit. The term was introduced by Rudolf von Kolliker in 1863, originally as a synonym for protoplasm, but later it has come to mean the cell substance and organelles outside the nucleus. 3. 4. There has been certain disagreement on the definition of cytoplasm, as some authors prefer to exclude from it some organelles, especially the vacuoles. 5. And sometimes the plastids. 6. Physical nature, edit. It remains uncertain how the various components of the cytoplasm interact to allow movement of organelles while maintaining the cell's structure. The flow of cytoplasmic components plays an important role in many cellular functions which are dependent on the permeability of the cytoplasm. 7. An example of such function is cell signaling, a process which is dependent on the manner in which signaling molecules are allowed to diffuse across the cell. 8. While small signaling molecules like calcium ions are able to diffuse with ease, larger molecules and subcellular structures often require aid in moving through the cytoplasm. 9. the irregular dynamics of such particles have given rise to various theories on the nature of the cytoplasm. As a sol gel, edit. There has long been evidence that the cytoplasm behaves like a sol gel. 10. It is thought that the component molecules and structures of the cytoplasm behave at times like a disordered colloidal solution, sol, and at other times like an integrated network, forming a solid mass, gel. This theory thus proposes that the cytoplasm exists in distinct fluid and solid phases depending on the level of interaction between cytoplasmic components, which may explain the differential dynamics of different particles observed moving through the cytoplasm. A papers suggested that at length scale smaller than 100 nm, the cytoplasm acts like a liquid, while in a larger length scale, it acts like a gel. 11. As a glass, edit. It has been proposed that the cytoplasm behaves like a glass-forming liquid approaching the glass transition. 9. In this theory, the greater the concentration of cytoplasmic components, the less the cytoplasm behaves like a liquid and the more it behaves as a solid glass, freezing more significant cytoplasmic components in place. It is thought that the cell's metabolic activity can fluidize the cytoplasm to allow the movement of such more significant cytoplasmic components. 9. A cell's ability to vitrify in the absence of metabolic activity, as in dormant periods, may be beneficial as a defense strategy. A solid glass cytoplasm would freeze subcellular structures in place, preventing. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Proteins in different cellular compartments and structures tagged with green fluorescent protein damage, while allowing the transmission of tiny proteins and metabolites, helping to kickstart growth upon the cell's revival from dormancy. 9. Other perspectives, edit. Research has examined the motion of cytoplasmic particles independent of the nature of the cytoplasm. In such an alternative approach, the aggregate random forces within the cell caused by motor proteins explain the non-Brownian motion of cytoplasmic constituents. 12. Constituents, edit. The three major elements of the cytoplasm are the cytosol, organelles and inclusions. Cytosol, edit. Main article, cytosol. The cytosol is the portion of the cytoplasm not contained within membrane-bound organelles. Cytosol makes up about 70% of the cell volume and is a complex mixture of cytoskeleton filaments, dissolved molecules, and water. The cytosol's filaments include the protein filaments such as actin filaments and microtubules that make up the cytoskeleton, as well as soluble proteins and small structures such as ribosomes, proteasomes, and the mysterious Volt complexes. 13. The inner, granular and more fluid portion of the cytoplasm is referred to as endoplasm. Due to this network of fibers and high concentrations of dissolved macromolecules, such as proteins, an effect called macromolecular crowding occurs and the cytosol does not act as an ideal solution. This crowding effect alters how the components of the cytosol interact with each other. Organelles, edit. Main article, organelle. Organelles, literally little organs, are usually membrane-bound structures inside the cell that have specific functions. Some major organelles that are suspended in the cytosol are the mitochondria, the endoplasmic reticulum, the Golgi apparatus, vacuoles, lysosomes, and in plant cells. Chloroplasts. Cytoplasmic inclusions, edit. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Main article, Cytoplasmic inclusion. The inclusions are small particles of insoluble substances suspended in the cytosol. A huge range of inclusions exist in different cell types, and range from crystals of calcium oxalate or silicon dioxide in plants, 14, 15, to granules of energy storage materials such as starch, 16, glycogen, 17, or polyhydroxybutyrate, 18. A particularly widespread example are lipid droplets, which are spherical droplets composed of lipids and proteins that are used in both prokaryotes and eukaryotes as a way of storing lipids such as fatty acids and sterols. 19. Lipid droplets make up much of the volume of adipocytes, which are specialized lipid storage cells, but they are also found in a range of other cell types. Controversy and research, edit. The cytoplasm, mitochondria, and most organelles are contributions to the cell from the maternal gamete. Contrary to the older information that disregards any notion of the cytoplasm being active, new research has shown it to be in control of movement and flow of nutrients in and out of the cell by viscoplastic behavior and a measure of the reciprocal rate of bond breakage within the cytoplasmic network. 20. The material properties of the cytoplasm remain an ongoing investigation. A method of determining the mechanical behavior of living cell mammalian cytoplasm with the aid of optical tweezers has been described. 21. See also, edit. Amoeboid movement mode of locomotion in eukaryotic cells. Cytoplasmic streaming flow of the cytoplasm inside the cell. Protoplasm, alternative term for cytoplasm or cytoplasm and nucleoplasm. Syncytium type of multinucleate cell. References, edit. 1 Shepherd VA, 2006. The cytomatrix as a cooperative system of macromolecular and water networks. Current topics in developmental biology. Volume 75. pp. 171223. Do. 10.1016/S0070-2153, 06, 75006. 2. ISBN 9780121531751. PMID 169848813. 2 Hogan cm, 2010. Calcium. In Jorgensen, Cleveland C., eds. Encyclopedia of Earth. National Council for Science and the Environment. Archived from the original on 12 June, 2012. 3 von Kolliker A., 1863. 4. Auflage. Handbuch der Jubiläre des Menschen. Leipzig, Wilhelm. Engelmann. 4 Bynum W.F., Brown E.J., Porter A., 1981. Dictionary of the History of Science. Princeton University Press. ISBN 9781400853410. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 5 Parker J., 1972. Protoplasmic resistance to water deficits. In Kozlowski T.T., ed. Water Deficits and Plant Growth, Volume 3. Plant Responses and Control of Water Balance. New York, Academic Press. pp. 125176. ISBN 9780323153010. 6 Strasburger E., 1882. Je Bedentheilungsvorgang der Zelken und das Verhältnis der Kernteilung. Zellteilung. Archmika Annett. 21, 476590. Do. 10.1007/BF02952628. HDL 2027/HVD.320441061999177. S2 SID 85233009. Archived from the original on 27 August 2017. 7 Cohen, Mora U2, Skuf JC, Slepchenko BM, Lo ULM, 2012. Spatial Modeling of Cell Signaling Networks. Computational Methods in Cell Biology. Volume 110. pp. 195221. Do. 10.1016/B978. 0-12-38840-3-9.00008-4. ISBN 9780123884039. PMC 3519356. PMID 2248290. 8 Holtmann D., Karenbrot J.I., April, 2004. Longitudinal diffusion in retinal rod and cone outer segment. Cytoplasm, the consequence of cell structure. Biophysical Journal. 86, 4566-82. Bibcode, 2004 BPJ, 86.2566H. Do. 10.1016/S0006-3495, 04, 74312X. PMC 1304104. PMID 15041693. 9 ABCD Parry BR, Suroft Sev 4, Kabi N Mount, Ahern CS, Dufresne, Jacobs Wagner C., January 2014. The bacterial cytoplasm has glass-like properties and is fluidized by metabolic activity. Cell 156, 1218394. Bibcode 2014 APS March 16002P. Do. 10.1016/J.Cell.2013.11.028. PMC 3956598. PMID 243611104. 10 Taylor CV, 1923. The contractile vacuole inuplets, an example of the sol-gel reversibility of cytoplasm. Journal of Experimental Zoology. 37, 3259289. Bibcode, 1923 JES, 37259T. Do. 10.1002/Jes.1400370302. 11 Quapisiuska, Karina, et al. 31 July 2020. Nanoscale viscosity of cytoplasm is conserved in human cell lines. The Journal of Physical Chemistry Letters. 11, 16 6914 6920. Do. 10.1021/X.Chplet.0C01748. PMC 7450658. PMID 327872003. 12 carat Guillaume, Ulrika H., Jensen M.H., Renz M., Moore Jr., Goldman Road, Lippincott Schwartz J. Macintosh F.C., Whites the, August 2014. Probing the stochastic, motor-driven properties of the cytoplasm using force spectrum microscopy. Cell 158, 48222832. Do 10.1016/j.cell.2014.06.051. PMC 41830665. PMID 251268787. 13 carat Vans Honor, Mosin Kemh, Skipper RJ, Sonneveld P., Weimer EA, September 2003. The Volt Complex. Cellular and Molecular Life Sciences. 60, 9/18/2837 do. 10.1007/00018-003-3030 PMC 11138885. PMID 145235546. S2 SID 21196262. 14 carat Prikid, Christina J. Rudel, Paula J. 1999. Calcium oxalate crystals in monocotyledons, a review of their structure and systematics, PDF. Annals of Botany. 84, 6725739. Do. 10.1006/anbo.1999.0975. 15 carat Prikid CJ, Rudel PJ, 2004. Systematics and Biology of Silica Bodies in Monocotyledons. The Botanical Review. 69, 4377440 10.1063/0006-8101, 2004, 069, 0377 Sabasp, 2.0.co, 2. JSTA 4354467. S2 SID 2452043. 16 carat Ball SG, Morel MK, 2003. From Bacterial Glycogen to Starch, Understanding the Biogenesis of the Plant Starch Granule. Annual Review of Plant Biology. 54, 207233. Do. 10.1014/anav.rplant.54.0319002.134927. PMID 1450290. 17 carat Shira J. Graham Ter, April 2002. New Perspectives on the Storage and Organization of Muscle. Glycogen. Canadian Journal of Applied Physiology. 27, 217920 do. 10.1139/H02-012. PMID 121799957. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 18 carat Anderson H. Dawes EA, December 1990. Occurrence, Metabolism, Metabolic Role, and Industrial Uses of Bacterial Polyhydroxylketones. Microbiological Reviews. 54, 445472. Do. 10.1121.24.4.450-472.1990. PMC 372789. PMID 2087222. 19 carat Murphy DJ, September 2001. The biogenesis and functions of lipid bodies in animals, plants and microorganisms. Progress in lipid research. 40, 5325438 10.1016/S0163-7827, 01-00013-3. PMID 11470496. 20 for Nieberg W., Westphal M., Sackman E., August 2001. Dictyostelium cells cytoplasm as an active viscoplastic body. European Biophysics Journal. 30, 428494 do. 10.1007/S0024901001001335. PMID 11548131. S2 SID 9782043. 21 Hu J, Jafari S, Han Y, Grodzinski H, CAIS, Gyo M, September 2017. Size and speed dependent. Mechanical behavior in living mammalian cytoplasm. Proc. National Acad. SCI. USA 114, 36 9529. 9534 bibcode, 2017 as 114.9529H. Do. 10.1073/NAS.17024888114. PMC 5594647. PMID 2882733. External links, edit. Lubefeld K. 2000. Site Architecture and Physical Properties of Cytoplasm, Volume. Viscosity, diffusion, intracellular surface area. Microcompartmentation and phase separation in Cytoplasm, PDF. International Review of Cytology. Volume 192. pp. 189221. Do. 10.1016/S0074-7696, 08, 60527-6. ISBN 9780123645968. PMID 10553280. Archived. From the original, PDF, on the 11th of September 2008. Citebook, Journal =ignored, help. Category, Cell Anatomy. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.